And here we go with chapter 4. This image um, at the beginning of chapter 4 is stunning. And it's relatively new technology. And often when I see these the first time, I think it's an artist rendition of what's true. But these are axons coursing through the human brain. And we can image them and map them. We are creating a connectome, and it's stunning. It's stunning. My head spins to think about what we'll be able to do in 50 years. You'll have to let me know. Okay, methods of research in psychopharmacology. Um, I'm a behavioral neuroscientist. I'm a psychologist. I do behavior, and it is my strength. And the first third of this chapter is about behavioral assessment of drugs and such. And so, I can contribute more there, and I will. The last third of the chapter is all the bleeding edge new technologies we have. Largely, I haven't used them. I read about them, but I haven't done them. And the book does an excellent job of covering them, so I will have to leave that largely to the book, other than to continually express amazement. Sadly, in this chapter, it opens with a mistake. Not that we may have 100 trillion synapses or 100,000 miles of axons in the human brain. It's been estimated that there are more potential neural states in the human brain than there are atoms in the known universe. Just run that around in your head for a while, because each synapse can have multiple states. That's all true. And but the red word in the middle of this opening vignette is a mistake. It's not until the Spaniards, Golgi and Cajal, began to stain neurons. Hello. You might want to look that one up, which I have done, of course. So here's the 1906 Nobel Prize for Physiology or Medicine. And there's my good friend. I wish I were his good friend, Cajal on the right. And there's Golgi on the left, who I can barely relate to. Let's click on Golgi and see what it says about his country of origin. Oh, Italy. Born in Italy, died in Italy, therefore a Spaniard? I don't think so. So that's a mistake, not a big deal, but it's a mistake. Oops, that's spoiler alert. We'll come back to that. Connectomes, Human Genome Project, it's just stunning. We have now mapped all the neurons and connections in some simpler organisms. Our ability to understand behavior is getting better and better. So again, my thing, I'm a behavioral neuroscientist, and techniques of behavioral pharmacology, that's me. Okay. On the lower left-hand side, you're going to see the terms in vivo and in vitro all the time. Just spend a little time with that sentence, and learn how to use those Latin phrases accurately. It's important. You are, if I do something to you, that's in vivo. You are a living organism. But if I take a piece of you and put it in a dish and do something there, that's in vitro. Okay, we're going to deal with some research methods terms here. I spent a lot of time teaching research methods. The issue of validity and reliability is a very big deal in any kind of science. You can measure some things very reliably, which basically means over and over and over and get the same result. but they may not validly indicate what you think they do. So reliability and validity are very important concepts here. Basically, you can't be valid if you're not reliable, but you can be reliable as heck and not be valid, as we'll see. Ode to animal research. This is an emotional issue, there's no doubt about it. Um, Probably some of you in the room are alive because of animal research. And you have every right to be ethically involved in or ethically challenged by this. And it is a moving target. Um, I found my own positions on things to have changed a bit. I have done a lot of animal research in my day. Correlational relationships. Um, Particularly with human disease states and the relationship between salt intake and hypertension or chemical exposure and cancer. Those are correlational relationships, and they have every right to be in science. But I'll tell you that if you're going to be a researcher dealing with correlational data, you have to be wicked smart. To do an experiment, you have to manipulate a variable. If you don't manipulate a variable, you're not doing an experiment. Do we know whether smoking causes health problems in humans? Well, we haven't done an experiment because it's unethical, not because it's hard to do. But have we made people smoke to see if they get cancer? Will we ever make people smoke to see if they have cancer? No, because of ethics. So we've had to make conclusions about humans based on human correlational data. Have we made animals smoke? Yeah. And have there been health consequences, negative ones? Yes. So we have demonstrated a causal relationship through experimental research for health-related things in animals, infrahuman animals, or animals too, um, that we haven't done in humans. And big tobacco, and now big sugar and big alcohol, uh, they want you people confused. Uh, Big tobacco used to pay PhDs to go in front of cameras and say, a causal relationship has never been demonstrated between smoking and negative health benefits in humans. And I can say that to this day. It hasn't been demonstrated because an experiment hasn't been done, because it's unethical, not because it's confusing. A causal relationship between the moon and the Earth's tides has never been demonstrated. Does that mean I wouldn't bet my life or your life on the moon affecting the Earth's tides? I am positive, to the extent a scientist can be positive without experimental evidence, that the moon causes changes in the Earth's tides. But if we want to use the gold standard of doing an experiment, we have to move the moon. We have to blow up the moon and see if the Earth's tides change. Good luck with that. Some of the smartest researchers there are not doing experiments. They're doing correlational research. It's wicked hard, and probably the majority of adult humans cannot even think about things um, that are as complicated as correlational research. As I love to say, any idiot can understand an experiment, but boy, correlational research is tough. There's a few of these lists in this chapter. I, for decades, was the chair of our school's animal care and use committee, and I took that position very seriously. And there are guidelines. The federal government cares about this very much. There are surprise inspections where veterinarians come in and literally do white glove inspections of the animal facilities looking for dust. It's very real. Now, face validity. I have a colleague who, when he teaches about psychology and validity, he says he had me deliver one of his lectures, and he writes everything out. And it said in there, face validity, turn to the side and spit, is no validity. That means it just looks good. It looks like it's measuring what it should, which may mean it is, but you can't trust it. Predictive validity, construct validity, there we go. So will it predict future outcomes? Is it really studying the construct, the concept that the investigator thinks it's studying? And here's another list. As I said in the previous chapter, I remember having to memorize those criteria for whether something was a neurotransmitter. I had to memorize these. I think I put them in part of my dissertation. That if a behavioral test is going to have good predictive validity, one, it should be specific for the class of drug being screened. Two, it should be sensitive to, so that the doses used are in a normal therapeutic range. And three, it should demonstrate the same rank order of potency, ranking of drugs according to dose efficacy. In the early days of studying opiates and whether behavioral tests for pain, which is coming up here shortly. These were critical, and researchers very obsessively did this research to assess whether their tests were valid. And of course, if it's not reliable, if the data are all over the place, when you rerun the assessment, you're done. You're not going to get anywhere with that. Simple behavioral observation, catalepsy. I may have mentioned the video I made of the animal that I gave an overdose of morphine to demonstrate the effects of naloxone. They became cataleptic. They were immobile. And some researchers, would record that and measure how long it took for them to start moving again. And some antipsychotic drugs produce those effects. Some antipsychotic drugs used to be called zombie drugs because when you took too many of them, you sort of looked like the Frankenstein monster, or Frankenstein's monster. Stiff, masked, face, no emotional expression, slow movement, robot-like movement. Those, that's good research. The open field test I've used many times. Personally, I like my open fields test environments round, so there are no corners, because corners are magnets for fearful animals. They just stick in the corner. But unfortunately, a lot of researchers now use square open fields. I still like them round. And a nervous rat or mouse, they defecate. And one indication of fear in an open field is pellets. And they also urinate. And you can make the urine glow and count the spots. So there's often multiple ways to assess the same concept. And a fearful animal will stay up against the wall of an open field. They won't go into the middle. And a fearless animal will wander all over the place. Operant conditioning, this is what I did in grad school a lot. It's used in psychopharmacology. I would say it's not used a ton, but when you think of operant conditioning, you might think of B.F. Skinner, because that's appropriate, who briefly lived in Scranton, PA, and his brother is buried here. Med Associates is one of the major manufacturers of behavioral testing units today. And if you're wondering what we're looking at here, other than the rat, these little spots here, each of those is for a rod that goes across the bottom of the enclosure. They can be electrified. This over here contains pellets that no doubt the animal is consuming. There are multiple potentials for cue lights and sounds, and there's also often the option of giving the animal water. So you can do a lot with that. Schedules of reinforcement. I have a whole lecture on this, and the book, samples a few of these schedules. Fixed ratio. It's a ratio. It's a number, and it's fixed ratio. And it specifically mentions FR3, but then it mentions, well, the ratio can be changed across time to higher and higher numbers. And pharmaceutical companies use this a lot to assess how much animals like drugs. They ask them to do more and more lever presses to get their reinforcement. And they look for the breaking point beyond which the animal says, this isn't worth it. And now we get to my stuff, measures of analgesia. I showed you briefly an abstract from an article I did long ago on stimulation-produced analgesia, where we electrically stimulated the midbrain of rats and found two descending brain to spinal cord pain inhibitory systems, one opioid, one non-opioid, and the non-opioid was endocannabinoid, your brain's own marijuana. The behavioral test we used in that was the tail flick test. This picture here is a bit imaginary. The animal has to be gently restrained, and I do mean gently. When I came to Pennsylvania, I used soda bottles, which were not available in Pennsylvania. I used to drive across the New York border to buy 1 liter bottles of soda, which were not available here for a while. And the light source, if you've ever seen an old projector, slide projector, or an overhead projector, we use those bulbs. And rheostat varies the voltage to the bulb. So you adjust the heat of the bulb to get a baseline of a certain amount. This is one heck of a baseline. Oh no, well, I like my baselines at around 3.5 seconds. They're going much longer. And usually your light goes off automatically at double the baseline, and you would call that 100% analgesia. It's a good little test. It's spinal. You can literally cut the spinal cord, and after the shock of the cut is over, the lower spinal cord has its own blood supply, and that tail flick response will keep going in the lower spinal cord. In fact, it will get bigger because there's brain to spinal cord modulating systems that make it a little bit muted. In fact, when physicians do the patellar reflex, they put a mallet on your knee. It's a bad sign when that reflex is too big, because that suggests that there's a disruption of descending brain to spinal cord modulation of that reflex. So that's a very common measure of analgesia. It's a spinal reflex, and what's neat about it is to change it. Presumably, you have influences going from the brain to the spinal cord, unless you're giving a drug that goes everywhere. Hot plate, I've used that less. Often, I have a big block of aluminum. It floats in water at a set temperature. I think they mentioned Yeah, between 55 and 70. Usually you only use one temperature. The animal is, there's a plexiglass cylinder that's way higher than the animal can jump. You put the animal in there and you wait until they jump or they thump their back foot, and then you take them out. That behavior is not spinal. It involves brain mechanisms up through the thalamus and potentially even beyond. So with the tail flick and hot plate, you can look at pain, reactivity and pain inhibition at the spinal level and at the upper brain level. Radial arm maze. I think I have a video of this for you. In the early days of psychology, researchers used these to study learning, just straight learning. No neuroscience really need apply. And when I went back to UCLA one summer and I walked into a room that used to have an office in it, there was one of these in the middle of the room. And I thought, geez, this is historical. This is something we don't use anymore. Well, boy, was I wrong. This is now a very common measure of spatial memory. Notice the plexiglass is clear, so the animal can look around. And of course, they can see where they are when they go down the alleys. So it's a spatial learning task, and you can assess what's called working and reference memory. Working memory is the animal should remember it just ate that food. And if it ate the food, it shouldn't go back and eat it again. Reference memory is during training, only some of these arms will be baited, and the animal learns to only go down the arms that has food. Let me find you that video. Oh, I think I put this. I know I put this on your course homepage, which is right. It will be right here. So here's radial arm maze post-training knockout mouse. So later in this chapter, there's talk about knockout mice, which we haven't mentioned yet today, but by the end of the chapter, you'll know. And here's a knockout mouse, and there's accompanying music. I'll talk over it. So this animal has had adequate training. It should know what it's doing. Those are the armed. That's where the food is. And here comes the animal, and it gets a pellet right away, and that indicates the pellet has been. Okay, that's a mistake.
You may find yourself in another part of the world. You may ask yourself, well, how did I?
Sorry about that.
You may find yourself in another part of the world. You may ask yourself, well, how
The animal loves that arm.
You may ask yourself, how do I work this? You may tell yourself, this is not my beautiful house. You may ask yourself, where does that highway lead to? You may ask yourself, am I right? Am I wrong? You may say to yourself, my God, what have I done? Let in the days go by. Let the water hold me down. Let in the days go by. Water flowing underground. Making a blue again. After the money's gone. Once in a lifetime. Water flowing underground. Same as it ever was.
All right, let's pause that here. So notice they're talking about long-term errors and short-term errors. Long-term, that's reference memory. The animal should have known to never go down that arm at all. Short-term errors mean, hey man, you already did that. And if you've done it once, you should remember what happened there, and you shouldn't do it again. This is a genetic knockout mouse. Again, you'll understand that much better at the end of the chapter. And he has problems with both working and reference memory. And that link will be on, or is on, the page that you got to this lecture through. So that's radial maze. Multiple T mazes aren't used very often these days. Okay, I have a video of this too, and this is called the Morris Water Maze, and Morris is capitalized. That's a human being, and he gave a talk a couple years ago at Neuroscience. Must be amazing. This is one of the most popular measures of spatial memory in animals today. It's used all the time. It must be pretty amazing to be Morris and have the Morris Water Maze used all the time. So there's a big tub. We made one of these. It flooded the biology department's animal facility. All right, I didn't flood it. It just made it wet. I fixed it. In there is usually opaque water. They originally used powdered milk. And when rats come out of there, they do what dogs do when they come out of a lake or a bath. They shake, and they shake liquid powdered milk all over the place. It's disgusting. But notice the room. That room is pretty chaotic. There's a computer, there's a chair where the investigator will sit quietly. There's a clock, there's a triangle and a chessboard type thing, and a picture of pine trees. These are visual cues that you want the animal to use to navigate this. Notice that the walls between the water and the top are not that steep. So the animal can, look around this room as it's navigating. Let's see that one. So this is a video, and it's nowhere near as pretty as the video or the picture in the book. But this is a video of an animal on its first training day. More music.
With 25 million bookable rooms and hotel.
So we use non-toxic latex paint now, not milk. And that circle indicated where the platform is. It's just under the water, so they can't see it from up top. Notice how the animal is staying near the walls. Up now he's getting adventures. They swim really well, and he's swimming, or she is swimming right over the platform, and either they don't trust it, or they're so busy swimming their legs aren't sticking down and they're not feeling it. I don't know, you can see some of the cues. Now here we go. Now we're gently moving the animal to the platform. He's too freaked out. Gently moving the animal to the platform. He's too freaked out. In my world, we've done this one. The operator would gently hold them and put their hand in front of them to make them stay on the platform at first. Okay, so that's day one. Usually there's, and it says escape latency 2 minutes. Of course, they didn't escape. So this is 5 days later, 2 trials a day, trial 10. Watch this. Hello. The operator doesn't even get out of the room. So they learn that very well. So that's the Morris Water Maze. We have that. This is used quite a bit in memory research in primates, but it's a little bit rare to do research in primates, but it's well described. Just take a look at that. And done a lot of anxiety research. And this elevated, why is it called elevated? Because it is. Why is it called a plus maze? Because it is. We made one of these. Sometimes they have little ledges here and here. Sometimes they don't. But notice that this arm has no walls, and these arms have big tall walls. So try to imagine yourself as a scaredy cat, mouse, or rat. Where are you going to go? You're going to go where there's these nice walls all around you. And if you're fearless, you go out here and just hang out and look around. So it's a good measure of fear. Um, like dark crossing, animals, uh, rats and mice tend to like the dark. So for them to leave a dark, safe area and move into a bright lit area, that indicates they're pretty fearless. One chamber social interaction task. You put the animals together and see how they interact as it's described. That's pretty cool. On the next page is something I've also done. Whoops. Oh, the three chamber. We'll get to that. Novelty suppressed feeding. When something new is around, it can disrupt your feeding. Animals or rats and mice emit ultrasounds, meaning we can't hear them, but we have technology that can, and we can listen to those calls. The Vogel test, back in when I was you, that was very popular. It's not used as much now as it used to be, but it's a conflict. The water spout, which is made of stainless steel, is delivering water or often sucrose, and ooh, I want sucrose. But then you can wire it up where they get shocked for licking, and you have a conflict. I want the salt water, sugar water, but I don't want to get shocked. And so the Vogel test assesses that. Fear. Conditioned emotional response, often used or historically used in learning research. Pharmacology, a little less common, but it's well described. If you're freezing in fear, you're not engaging in operant behavior to get food, and the amount of responding or liquid, and the amount of responding you give drops off, so you can measure suppression of behavior. Fear potential startle. I would prefer that was called fear potentiated acoustic startle. I did my doctoral dissertation on acoustic startle. I love it. And the guy that I kind of was paralleling, Michael Davis, he kept going with acoustic startle, and he found modulating pathways, and fear is one modulation. But the book somewhere in here does talk about a pre-pulse where you give a sound before the loud acoustic startle stimulus, and usually that inhibits the acoustic startle because you're warned that you're going to hear the sound, but not some people. Drug intake can affect that, and schizophrenics who are unmedicated don't show that very well. And as you can imagine, fear, potentiated startle is can be very strong in PTSD patients. So I would prefer you call it fear potentiated acoustic startle. So you put an animal in a fearful situation. Well, you've seen this yourself, no doubt, when you're watching a scary movie. When you think something bad's going to happen and there's a sudden noise, you jump. That's fear potentiated acoustic startle. Okay. We'll come back to depression. I just want to mention this three-chamber social interaction test, which, as you can see, is one thing we use to look at social interaction, which seems to be related to autism spectrum type behavior. And here in the next page, is the figure I was looking for. And there's 3 chambers. You start in the middle, and you've got 2 enclosures. One is empty, or sometimes you put something in there that's inanimate. And over here in the cage, you have someone to interact with. Now, by the way, if you ever visit me in my office, I read the original research here and I followed their protocol and I bought these. And if you look at them long enough, you'll probably recognize there's foam on the bottom and they're wider at the top. They're pencil holders. They've got them upside down. And just as was the case with me, you put a mouse in there and it's, shiny metal on the bottom on a plastic base. The animal just starts pushing it around. So you have to put weights on them to keep the animals from pushing them around. But if you're, if you don't like people, in this case, mouse people, you're not going to spend much time interacting with a peer over here. So it's an interesting study. It works well. It's used pretty commonly. Okay, now we get into some stuff. I did have videos on these, but I choose not to show them. Depression is a very relevant condition to the human condition. And if we want to do studies on animals that are, related to depression, we need models, not just models that look good, but models that are actually related to the human condition. How do we know? Well, one thing, we can use drugs that affect depression in humans on animals in these conditions, in these tests, and see if the drugs work on animals in the way they work on humans. And these tests have been validated. So here's the forced swim test. By the way, I wouldn't allow that in my lab. Why? Notice that the animal's tail is touching the bottom of the container. I don't allow that. I make the water deep enough so the tail will not touch. But the animal's in water. Now, we saw animals in water in the Morse water maze. Why isn't that a measure of behavioral despair? Well, geez, the Morse water maze, you've just been thrown into an ocean and you've got all these places to explore, and you explore. When you do behavioral despair in the forced swim, you put the animal in a small enclosure so they can realize quickly, oh my goodness, there's nowhere out of here. The walls are slippery. The thing is not much bigger than me. I've explored everything. And the animals seem to give up. They don't give up and drown. That's not allowed. They float really well, by the way. But they stop struggling. They stop trying to swim. And what we measure is the amount of time they float without swimming. Now, they're only in here for, I forget, like 3 minutes, maybe 6 minutes. It doesn't photograph well, but in terms of, and often you only do it once to the animal. So there are worse things that can happen, but it is a valid measure of depression for animal research, and it is fairly widely used. There's another one that they don't talk about. Oh, there it is. They don't show it. The tail suspension test. This doesn't photograph well either. You go to your tape dispenser, 3M. You make a little flag of tape on the base of the tail, and animals can be handled very comfortably at the base of the tail. Tip of the tail, no. Base of the tail, very easy. And you clip that flag to a hook at the top of an enclosure, and there's the animal hanging from its tail. And just like the forced swim test, animals give up and they just hang there. And you measure the amount of time they just hang there, and it doesn't hurt them, and it's despair, and it's been validated using the same drugs that work on people. And again, it's 6 minutes, and usually you only do it once. This chronic, mild, unpredictable stress, that sounds like something we do to prisoners who are trying to get information out of. This would bother me. You keep doing things that aren't so awful over and over again. It doesn't read well. It's valid, and it's very important. I mean, if you've known someone with anxiety or depression or PTSD, we need treatments. Social defeat, maternal separation, which we do at the southern border. Sucrose preference. This is an assessment of anhedonia, without pleasure. The animals don't seem to like sugar water as much as they normally do. And antidepressants can help. This I've alluded to earlier. This is what drug companies do, and they use fixed ratios, which I think come up here again. So there's a cannula that goes often into the carotid artery, and it's anchored, and it's there for the long term. So this is like self-administration of a drug. The animal goes into the chamber. They're very used to this situation. They're not stressed. They're not in pain. And when they hit this lever, and we put them on different schedules, the infusion pump can give them their drug. We can do a lot of things with this situation. It is used by drug companies quite a bit. And here's what I mentioned earlier, the breaking point, when they just won't do it anymore. And it's a valid indication of the rewarding potential of drugs. And here are drugs that act as reinforcers there. By the way, I haven't checked lately, but my friend wrote a book on nicotine not being addicting. Yes, not. Smoking is a vicious habit that is very difficult to overcome, possibly more difficult than heroin addiction. But he claims that animals in this situation are not very prone to self-administering nicotine, but these other things, they will. Self-stimulation, I used to do that back in the day. It's still done. You have to do stereotactic surgery to put the electrode in the appropriate place, and you put screws in the skull, and you anchor it with dental cement, and it's a deal. And it is still sometimes done. Condition place preference. So you train the animal to associate a place with a drug. So you put them in the place and you give them the drug, and you let them experience the drug in the place. And then later, you give them a choice between that place and another place. And notice that the places are pretty different. The texture of the floor, the walls, there's often odor differences. And you ask the animal, hey, where do you want to be? Do you want to be where you had that drug before? Or do you want to be somewhere else? And if you like the drug, you tend to want to go back. And discrimination studies. This is one of the ways that we used to assess whether a new drug worked through mechanisms that were the same as known drugs. In other words, does this drug feel like that drug? And there's morphine historically my drug. But I never did a lot of this reward stuff. Again, I used morphine, opiates to inhibit pain, and naloxone to block the inhibition of pain. So there's a nice description of how to do that. And translational research, right? That's sometimes we're doing basic science and we're not doing it for clinical purposes, but a lot of what we do, we're trying to help with the human condition. This is a brilliant box. Federal Drug Administration. I have an FDA license, and my blood runs cold whenever they come to investigate me. But boy, getting a drug approved is a deal. Neurosurgeons, the only thing that controls them is lawyers. They get an idea and they go do it, at least historically. But boy, drugs. Getting a drug approved takes a long time. And sometimes it takes so long people get upset because they want the drug, they want to try it, not for fun, but for therapeutic purposes. So just appreciate how much time and effort and money goes into the drug approval process. It's a sorry, it's a deal. Okay. And there's another one, ADHD, which is an important mechanism. Read that. I've never used it. It's a good one. Some people can't stop. Okay. Stereotactic surgery, I've already alluded to this in terms of putting electrodes in the brain. The devices are expensive. We have them for people. People get electrodes implanted in them, and sometimes cannulae, which are tubes through which we can administer pharmaceutical products, or recording devices if we're trying to understand. Usually we don't leave recording devices in humans, but we certainly do in animals. Lesioning. When I was you, this is what I did. Hi, my name is Tim Cannon. I blow stuff up. So I would go into the nervous system with the stereotactic device and, make discrete lesions, which means destruction of discrete parts of the brain, and look for behavioral changes. In the olden times, we did it with basically heat. We would just, destroy the tissue, the cell bodies, the axons, everything. That's used much less now. I also did knives. I cut fibers of the auditory system with the knife. That was more focused. Neurotoxins are used more so today. And the neurotoxin can be focused on one cell type. And there's a lot of magical procedures that are about to start popping up in this that are, after my time. So there's the rat in a stereotactic. I've never used that exact design. The animal is anesthetized. Those are called ear bars because they are bars that go in the ears, and they anchor in the auditory canal. And the incisors go here and cut the scalp, drill a hole, lower your electrode, put in the screws that I mentioned before, and then cement it. And, it's the top of the skull. So the animals don't interact with that very much, and it's stable. So it shouldn't produce much discomfort. This is a stereotactic atlas. Looks like could be mice. That could be a mouse. Could be rats, a little hard to tell. But this is our road atlas. So these are millimeters up and down and in and out. And we want to go to this part of the brain, which is a reward area, it turns out. And we want to destroy it. We want to stimulate it. We want to record from it. That's what we do. And there's people. Oh my God. So here, these things go right in and clamp on the skull. Uh, I've never seen that done. I was offered the opportunity, but I knew I'd faint, so I didn't go. Cannula, there's a term I've used. Again, that's often a stainless steel tube through which things can be inserted. You can also get CSF, cerebrospinal fluid, from there. Intracerebroventricular, ICV. I did a lot of that back in the day. Neurotoxic. That was kind of new, and I didn't do that, but you can do it. And then, as I mentioned, specific neurotoxins that only kill certain neurotransmitters. Whew. This is complicated, and we do less of this than we used to, but you may have seen images of heart-lung machines where tubes come out of the patient and there are these rollers that move the blood through the tube because you don't want to have a pump literally touching the blood. So that's what this is, and that's what that is. And so what we're doing is we are pushing artificial CSF down and back up, and this tubing is semi-permeable at the tip, which allows some things to go through, and thus the term semi-permeable, like the cell membrane. And when the rat's brain is releasing a lot of dopamine, for example, the output tube is going to have dopamine in it, and it goes into these collection tubes, and we measure things, and off we go. There are more modern methods than this that allow for much greater temporal resolution. The temporal resolution here is not great. I mean, these tubes are only changed about once a minute or so, but now we have ways where we can look at neurotransmitter release at less than a second. So, and there it is, in vivo voltammetry. One of my peers did that in Australia, interestingly enough. And another one of my peers is using it in his research. He went to Penn State, Hershey, and did a postdoc. I think it was Duke, and learned how to do that. And he's doing great stuff with it. It's almost magical. HPLC, high pressure liquid chromatography. You're watching things go through beads and things of different sizes and charges go through at different times, and you get spikes. And apparently, if you know what you're doing, you can figure out what's going on. There's electrical stimulation, and I've referred to this many times. So there's your dental acrylic and your connector, and they don't show you the skull screws to keep everything nice and stable. And there's a human, and here you have things under the skin that can induction coils that can create electricity there. By the way, these could also be cannulae, and you could have reservoirs for drugs that could be delivered in there. These could be cannulae. They love juice, and they're looking at a screen, and they're just hanging out. They're gently restrained, like the rats. These are stable, and we do that. Patch clamp, that's fancy. Just you read it. Visual quantification. Okay, so this table tells you what's going to occur. radioligand receptor autoradiography. I've done a little bit of that. Immunocytochemistry, not so much. In situ hybridization, not so much. But there's a nice discussion of radioligand binding. Remember, this is an open book test, and there's references to concepts that will be nicely displayed, like Bmax and KD in the on the next page. Receptor autoradiography. When I just got into the field, when I was about you, they found opiate receptors, although they quickly started calling them opioid receptors in the brain. And they did it by doing autoradiography and, not originally, but eventually they did autoradiography. So this looks a little ogival. Bmax, that's when your receptors are saturated. So that gives you an indication of how many receptors there are. And KD, that looks like ED50. This is the red line is what you really see. These two functions interact. One is things going on, one is things going off. There's an autoradiogram, and this is cocaine. Monkey brain. All right, I'll trust you on that. But boy, there are some hot spots. And caudate and putamen are part of your basal ganglia. They're really hot. The amygdala, the amygdaloid complex, very hot. Notice this part of the cerebral cortex is not. But this part of the cerebral cortex is, where it's infolded. All right, so if it's infolded, it probably is a monkey. So remember the terms in vivo and in vitro. Now, hypocretin, I think, was mentioned earlier. So you develop antibodies for something, and blood with the antibodies is applied to the tissue, and immunocytochemistry tagged antibodies locate molecules within cells. So there we go, hypocretin. And the animal's brains have been extracted and sliced thinly and put on slides, and then the magic solutions are put on top and allowed to interact and then washed off, and it's amazing. It's amazing. So there's how to produce antibodies and immunocytochemistry. Again, more discussion of hypocretin. Radioimmunoassay. So here's a picture of what's going on there. Take a deep breath and walk yourself through that. You have labeled antigens, radio labeled antigen is added, but you can also have a known amount of an unlabeled antigen, and they interact, they fight, and depending on the fighting, a different percentage of the receptors will be bound. So here we go, quantity of unlabeled antigen. So as that goes up, the unlabeled outcompete the labeled. So the percentage of bound labeled, as you put more and more unlabeled in, it goes down. And oh boy, 96 well ELISA plate. It doesn't show you the pipettes that are used to put things in there. They're quite the pipettes. So this is going on in the labs at Scranton now, but after my time, in situ hybridization, I really can't improve on these discussions because I've never done them. But there's an hour. Hopefully we're getting close to the end. That is a rat or a mouse. Rat, and we're looking at heavy labeling in the basal ganglia. there, and probably also nucleus accumbens. So it looks, again, you slice them, you put them on slides, you put the magic substance on that, and you wait for things to interact, and you get pretty pictures, which you can quantify. And I like these because you can see the binding. You can see the cerebral cortex here has nothing. And this area in the middle, which I don't think has been mentioned in the book yet, that's the septal area. That is nothing. See these white spots here? Those are axons. And they're axons. And therefore, it's a bundle of axons, like the corpus callosum, which is up here. And they're not going to bind because it's binding in the soma. Genome-wide association studies. I don't do these, but I'm studying genes and the development of the face and the relationship between the development of the face and genes and things like bipolar disorder and homosexuality and intelligence. So genome-wide association studies are relevant to me, but I certainly don't do them. 2-deoxyglucose. Just as I was leaving UCLA, PET scans were the thing. And we couldn't do PET scans. That's later in the chapter. We couldn't do PET scans in rats and mice because they were too small. But we could do 2DG. I've done a little bit of that. CT scans. I'm not sure I've ever had a CT scan. They're murky. They're not high resolution. MRIs are much more common now, but they're used in humans, and most hospitals have them. Hospitals now are beginning to have MRIs. So here MRIs, they're the cool kids on the block. They're much higher resolution. They are certainly not in color. But when you do, sections reconstructed, they can look pretty amazing. And they're I've had several of these. When I was at UCLA, they were looking for lefties because they wanted to do MRI on the corpus callosum. And sadly, I am not a lefty. My dad was a lefty, and I generate lefty children like it's my job. But I was not invited for that study, which really still bothers me, obviously. Magnetic resonance spectroscopy is discussed here because you can do magical things with that related to Alzheimer's and Parkinson's and stuff. Diffusion tensor imaging, DTI, water in neural tissue. And this is where the amazing pictures of axons come from. This to me is still magical. I'm comfortable. Oh boy, I'm comfortable with MRIs and CT scans, and this is what I was trying to learn as I was leaving UCLA. There was a major grant, and they wanted UCLA to do research on this, and my lab was in on that, and then we got scared that we didn't know what we were doing. UCLA bought an inventor of this system and his technology. To do PET scans, you need to have a cyclotron down the hallway to generate the radioactive material. It's not very high resolution, but you can do amazing things with it. And now you can do specific neurotransmitters. I think I've seen a PET scanner behind where I go to get brain scans. Single photon, it's faster. And you also don't have to have a cyclotron. When you see these pictures, they often combine them with MRIs, and that's particularly true for fMRIs, which are down below. it looks like they're high resolution because there's a high resolution brain looking at you, but then there are these colored blobs. These scans are not high resolution. They are the colored blobs, but you often get an MRI in the same scan. So you reconstruct the brain with the MRI and then put the blob in place. This default mode network is probably important to depression and ruminating. Boy, I don't know if you know anyone with depression, but they often ruminate. They just think about negative thoughts, often about themselves, over and over. And by the way, a treatment that works for depression and even some forms of schizophrenia has nothing to do with drugs. It's cognitive behavioral therapy, and it can change your brain. But that's important. Personalized medicine. Someday we're all going to have our genomes read, and I had MRSA, and the original treatments for it, they didn't take into account that I'm a giant human being, and the drugs only worked for 20 hours, and for 4 hours every morning, the MRSA would come back. Gee, I wonder why. I'm giant. Give me more drugs. EEGs were very popular back in the day. This figure. I think this figure is slightly mislabeled. I did a search for the 1020 system, and I think if you look at that search and you'll see what the text refers to, the FZ, CZ, and PZ, they're in the figure. It's not a horrible mistake on the book's part, but they start mentioning things that I think are not there. This might be done in the psych department soon. Notice this peak here, N1. Notice this peak here, P1 and P2. N means negative, and P means positive. By convention, that's how we display them. I'm sure it made sense to somebody at some point. Those are event-related potentials, ERPs. We're almost there. Genetic engineering, how to make special mice, chimeric mice. One of my students went to UCLA and did this for years, and he never got the mice he wanted, and he left the field. CRISPR is huge. And it's opening up all sorts of new avenues for treatment. There was something about CRISPR in the BBC even today. Transgenic mice, optogenetics is huge. One of our graduates who went to Emory and then MIT, she does optogenetics. You see these pictures of usually mice with little glowing lights coming out of their heads. They have made channels go into the cells, specific cells in the brain that respond to light. And here's chloride, and also pumps. Read this and be amazed. My student was written up in a European news article as being a mouse whisperer because she was doing optogenetics and affecting reward systems in mice by making these receptors appear in specific neurons. It's so different than my day. I'm just going in and there we go. I forgot, right? There's a picture of that's the laser affecting the cells. And notice only some cells in the brain express the receptors. So you can flood this area with light, but only certain cells will respond. It's magic. And this is the beginning of your world. Transgenic model of Huntington. Be impressed by that. And by the way, beam walk. Um, I forget whether earlier in the chapter they mentioned Rotarod, but I've done this one. Making animals walk along a difficult beam and seeing how well they do and how well they swim. Um, there we go. There's the Rotarod. We have one of these in the basement. It rotates, and you can vary the speed. And don't worry, they only drop a little bit, and there's little micro switches down there, and there's chemogenetics and dread. I think we're almost out of here. Oh my God, this is magic. Magnetogenetics, magnets affecting things in the brain. You don't even have to implant stuff anymore. You can get these things implanted to implant themselves into cell membranes that respond to magnetic fields. Oh, I'm sorry. I made of that's this is Dart, but equally magical. And I think we are out of here. Right. I'm sorry. I got distracted by the Dart picture. Dart attaches a drug to a limited number of biochemically identified cells. Drug mechanism of action. This is magnetogenetics, which is different and not Dart. And there are your 25 questions. And I ran a little long again, which seems to be my nature. Have a good day.